Hello, everyone. Welcome to today's webinar. I'm Elaine Lim, contributing writer at CNN Media Group, and I will be moderating today's event. This webinar is titled timsTOF Pro and PASEF, Multiplying Sequencing Speed and Sensitivity in Mass Spectrometry-Based Proteomics, and is being sponsored by Bruker. CNN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. This presentation today is being sponsored by Bruker. For more than 55 years, Bruker has enabled scientists to make breakthrough discoveries and develop new applications that improve the quality of human life. Bruker's high-performance scientific instruments and high-value analytical and diagnostic solutions enable scientists to explore life and materials at molecular, cellular, and microscopic levels. During the presentation, we will hear from Andreas David Brunner, who is a PhD student in the group of Matthias Mann at the MaxQuant Institute of Biochemistry. He received his Master of Science in Molecular Biotechnology from the Technical University of Munich, which included a research stay at Harvard University. His research focuses on the implementation of advanced mass spectrometry instrumentation for proteomics and peptidomics applications. We will also be hearing from Florian Meier and Professor Jürgen Cox. Florian Meier is currently a PhD student in the group of Matthias Mann at the Max Planck Institute of Biochemistry. He received a Master's of Science in Chemistry from Saarland University with research stays at the North Carolina State University and the University of Quebec at Montreal. His research focuses on mass spectrometry instrumentation and novel acquisition methods for quantitative proteomics. And Professor Jürgen Cox heads the Computational Systems Biochemistry Group at the Max Planck Institute of Biochemistry in Martinsried. He received his PhD in physics at the Massachusetts Institute of Technology in 2001 and has since been instrumental in developing MaxQuant, a quantitative proteomics software package designed for analyzing large mass. Dr. Cox is the author of numerous peer-reviewed in the field of data analysis in mass spectrometry and quantitative proteomics. I will now hand over to Florian.
Yeah, hello everyone, and thanks for joining our webinar on the timsTOF Pro and PASEF, which we use to multiply sequencing speed and sensitivity in mass spectrometry-based proteomics. We have divided the talk in 3 chapters. So Florian, it's me, will start with a background on the technology and how the PASEF method was developed. Then Jürgen will take over and give you an introduction to MaxQuant and how it handles the 4-dimensional feature space by the timsTOF. And Andreas will finish with applications of the timsTOF Pro for high-speed and high-sensitivity proteomics. So we'll start off with the technology part. But first of all, what is this technology good for? And we use it for systems biology and in particular proteomics. So why are we interested in proteins? It's because they simply control all the cellular processes in our bodies. The proteins are involved in gene transcription, in translation. They're involved in energy conversion. Proteins can be tagged by ubiquitin for degradation. Proteins can be used to transduce signals, and smaller proteins are also secreted and affect thereby the behavior of other neighboring cells. To study this super complex system, we used MS-based proteomics technology, and every MS-based proteomics workflow starts with a sample preparation, where the goal is first to extract the proteins from our cell, tissue, or body fluid. once we have achieved that, we typically digest proteins with proteases like trypsin or lyc, and we end up with a complex mixture of tryptic peptides, which we, in the next step, separate via liquid chromatography. And the cool thing is that we actually, now the technology developed so far that the sample preparation can be done in a single vial, or in this case shown in a stage tip by the in-stage tip method introduced by Niels Kulak and Matthias Mann a few years ago. And this now allows us to automate and parallelize sample preparation and scale this step up. And once we have those peptides cleaned and prepared for liquid chromatography, the mass spectrometer comes into the game. And here we typically use high-resolution mass spectrometers to be able to distinguish all the thousands and millions of eluting peptide features. And we separate them on columns that look like these. So everything is based on nanoflow chromatography, and we typically use 50 centimeter long columns with a 75 micron inner diameter, and they are packed with 1.9 micron beads with a C18 coating. And then the mass spectrometer performs two types of experiments. One is the so-called full scan, which is used to acquire the intact masses of the peptides as they elute from the chromatographic column. And the second experiment is the so-called MS scan or MS2 scan, in which intact precursor ions are fragmented into smaller pieces, and these are then computationally used to identify first the peptide and use this information to infer the protein identity. And we typically use this with the MaxQuant software that was developed by Jürgen Cox in our group here. And you will hear later on in the second part of the talk more about this and how this is adapted to the timsTOF. So as you can already guess from this workflow, proteomics is first of all a super complex technology. So what you see here is the output of the MaxQuant viewer, and every colored dot represents a single peptide feature in the MOZ and retention time space. As you can see in the zoom in panel B, there's a lot of features or peptides co-eluting. They have very similar masses, and at any retention time there is hundreds of them eluting. And what's a bit shown in panel C is that they have highly different abundances, and for any mass spectrometer that's super challenging to detect as many and as accurate as possible these peptides. So you can guess, in this case, we only have the liquid chromatography and the mass as separation dimensions, and any dimension of separation that we add to this can be very helpful. One promising field there is ion mobility spectrometry. To give you an impression of what ion mobility spectrometry can achieve, there is a little example of two peptides. So just imagine they have the very same mass, but it looks already as if they are folded a little bit different. We then let them collide with neutral gas molecules like nitrogen. We end up with so-called collisional cross-sections. And as you can see here, the peptides have the exact same mass, but one of them is a bit more folded than the other one, so they end up having the same mass but different ion mobilities. So there's different devices and technologies that are used to separate ions based on these properties, but our favorite tool to separate ions by ion mobility is the so-called trapped ion mobility device, which was developed by Mel Park and colleagues already back in 2011. It was first described. And the big advantage of the TIMS tunnel is that it's a very compact device. So the entire separation takes place in a TIMS tunnel, which is about 10 centimeters in length. And then there's two opposing forces. One of them is the drag of the gas flow coming in from the ion source. And opposed to that, there is an electrical field gradient that pushes the ions toward the entrance of the mass spectrometer. So this is shown here by the two arrows annotated with a VG and E. And once we have this, we can accumulate and trap the ions in the TIMS tunnel. And as a function of these two forces, they will separate along the axis of the TIMS tunnel. And low ion mobility ions, which is here visualized by the big red ball, they will come to a rest closer to the mass spectrometer, whereas more mobile ions will come to a rest at a position closer to the entrance of the TIMS device. And then we can sequentially elute the ions as a function of their ion mobility by lowering the electrical field gradient. So first, the red ions will elute, and then in the next step, the yellow ions will come out, and then the green ones, and then the very last ones will be the very mobile blue ions. And as they elute from the TIMS device, we can detect them with a mass spectrometer. In our case, that's a quadrupole time-of-flight instrument. To give you an impression of the separation power of the TIMS device, here are two small examples of synthetic peptides. On the left, you see two peptides that have the exact same mass, but a scrambled sequence. So they are not distinguishable by the mass spectrometer alone, but if we turn on the ion mobility device, we can nicely separate them as a function of their collisional cross-section. Also, small changes to the peptide already can have an impact on the ion mobility, which is shown here on the right. So it's the very same peptide sequence, but in the green case, we added a phosphor group to the peptide. And what happens is that the phosphopeptide is actually more mobile than the non-phosphorylated peptide. So we can do this with these direct infusion examples of synthetic peptides. But with a timsTOF Pro, we can now also do the same thing on a global scale for many, many peptides that we detect with liquid chromatography and LC-MS-based proteomics workflows. And what we found in our hands is that one of the key features of the TIMS device is that it's highly reproducible. So what you see in the left panel is the reproducibility of several thousands of collisional cross-sections measured from peptides, 2 replicate injections. And you can see that the Pearson correlation was well above 0.99, and the coefficient of variation between replicate injections is typically below 1%. So we can use this ion mobility information as kind of a cross-section ruler, then easily define the collisional cross-sections of tens of thousands or 100,000 of peptides. I think this is an entirely new dimension for MS-based proteomics, and we just started to endeavor into this area and learn much more about the gas phase behavior of peptides and how they can be separated as a function of their collisional cross-section. Another topic I wanted to discuss is the so-called parallel accumulation serial fragmentation method. And this also makes use of the trapped ion mobility spectrometry device. And in this case, we use the TIMS device to multiply the sequencing speed or the sensitivity of our MS-MS events. So as I mentioned before, MS-MS events are important for us to identify peptides based on their fragmentation pattern. And if you just do a standard TIMS MS-MS experiment, you would accumulate the ions in the TIMS device, elute them as a function of their ion mobility, and then the analytical quadrupole would select one ion of interest, which is then transferred into the collision cell, and we acquire the MS-MS spectrum in the time-of-flight analyzer. So in this example here, the quadrupole would be selecting the green ion, and we would end up with the MS-MS spectrum of the green ion. However, this would take as long as the accumulation and ramp time of the TIMS device is. And in this case, this would have been 50 milliseconds. So we would have spent to acquire just a single MS-MS spectrum of this green ion. So, but hopefully you can appreciate that we can now use the sequential elution of ions from the TIMS device to select more than one ion in a single TIMS scan. This is what we call the PASEF principle. And the idea is that the quadrupole quickly jumps from one MRZ to the next MRZ as the ions elute from the TIMS device. So instead of selecting one green ion for the entire TIMS scan time, we would now quickly jump in between the masses, and in this way select multiple precursors in a single scan. So in this example, we would set the quadrupole to isolate the red ions as they elute from the TIMS device, which takes about 2 to 3 milliseconds. And once this ion mobility peak is gone, then we would quickly switch the quadrupole to the yellow ions for another 2 to 3 milliseconds, and then jump to the green ones, and finally end up with the blue ones. So it's important here to realize that the number of ions that we detect in the MS-MS spectrum is actually the same in all the cases. So as compared with the TIMS-MS mode, we do not lose any ions because they are compressed in narrow ion mobility peaks rather than continuously detected. So these slides here show a typical MS-MS experiment where we select precursors from the full scan, we isolate them with the analytical quadrupole, and fragment them in the collision cell. So as you can see here, we have nicely separated MS-MS spectra as a function of the ion mobility. And if you select a single precursor or a single MS-MS spectrum, it will exactly look like a standard MS-MS spectrum. But now, instead of having just one MS-MS spectrum, you have 4 MS-MS spectra acquired in the very same time. So we've described this principle back in 2015, and it took us now more than 2 years to implement this in a fully fledged based proteomics platform, which is the Bruker timsTOF Pro. The timsTOF supports this PASEF scan mode fully and automatically selects the precursors in all the dimensions. And there's been a lot of technological innovations going into this development. One of them was to, first of all, select the precursors with an algorithm in real time. The other one was how to set the quadrupole fast enough so that we can actually sequence as many peptides as possible in a short time. And the instrument also features a dual TIMS funnel, which allows to operate the instrument at a duty cycle close to 100%. So coming back to the feature selection algorithm, or maybe one addition here. So all of this works because of the time scales are nicely fit into each other. So if you think about a MS-based proteomics experiment, the LC time scale is in a range of minutes. The TIMS separation takes place in about 50 to 100 milliseconds, and the quadrupole jumps within less than 1 millisecond, and acquiring a full resolution scan on the TOF instruments takes about 100 microseconds. So everything fits nicely together. And one big hurdle on the development was the precursor detection algorithm. So this is actually how it was back in 2015. We were manually targeting precursors based on printouts, and then put them into the instrument control software. So luckily this has now changed, and everything is done automatically live with the chromatographic separation, and the algorithm optimizes the precursor placement in multiple PASEF scans. So this can, for instance, be used to select and fragment peptides separately that would not be separated by mass. In this case, this is just a screenshot from an LC-MS run of a HeLa digest, and you can appreciate that the precursors A and B are very close in mass. They're super low abundant, but still the precursor scheduling algorithm detected both of them, and they were separately fragmented within the LC with the following PASEF scans, and we ended up with 2 MS-MS spectra of reasonable quality, and they were identified and annotated by the MaxQuant software. because of this PASEF principle, where we can have multiple precursors in a single TIMS MS scan, we can multiply our sequence speed by the number of precursors that we can put into a single TIMS scan. So back in 2015, we extrapolated data from another QTOF system, and we said, okay, if we would end up with like 10 PASEF precursors per TIMS scan, we could actually multiply our sequencing speed by a factor of 10. And if we now look back at the data that we acquire in our lab, we actually find that we reached this goal that we predicted back then. Well, using 100 millisecond TIMS ramp, we end up with about 14 precursors per TIMS ramp, which translates into an average sequencing rate above 100 hertz over the entire gradient. And this is data from a human cell lysate. So to summarize the developments and the features of TIMS and parallel accumulation serial fragmentation, there's probably 4 key aspects. So the first of them is that TIMS enables us to separate precursors based on their ion mobility in addition to the mass separation and the expansion time separation. Also, since we accumulate the ions, we compress the entire signal into narrow ion mobility peaks, which increases the signal-to-noise ratios. And by the TIMS separation, we get the full precursor mass resolution for the MS spectra. So there is no multiplexing. Spectra are nicely separated and can be separately read out and identified. And all of this happens with multiplied sequencing speed, which can partially use to also increase the sensitivity. With this, I would like to hand over to Dr. Jürgen Cox.
Yes, so good afternoon, everybody, or depending on your location, good morning or good night. So we will be slightly switching gears now. So the next 15 minutes, I will talk about how do we now actually analyze the data that we get with the machine and methods that were described by Florian Meier in the previous part. And so for that, we are using the MaxQuant software, and we actually have been collaborating with Bruker for a long time, for the past 2 years, to actually accommodate the data that has this one additional dimension incorporated to accommodate the analysis of this data in the MaxQuant software. So maybe to start, let me just remind people what MaxQuant is. So this is a software for the analysis of shotgun proteomics data, which we have been developing since 2007, and since we have this very long period for scientific software of continuous development, it is by now a quite mature, efficient piece of software which has a large user base. So probably, at least by all the measures that are available to us, it's the most used proteomics software tool in the community. And so the license model is that there is none. So it's really available for everybody, which also includes companies who can make use of MaxQuant and also of the enhanced features that we have integrated now for the timsTOF Pro without any charge. And so probably a good proxy for usage of the software is probably the number of citations of the original MaxQuant paper, which actually then counts how often was actually the software used and helped the lab or the person to finish their research project with success. And we are just this year hitting the 2,000 citations per year limit, which is actually far away from any other software package in proteomics. So I also want to briefly mention that it's actually 2 softwares that we're developing in my lab. So it is the MaxQuant software and the PASEF software. So the sharing of work here is that the MaxQuant software does the processing of the mass spec signals, so starting from the raw data and ending with the identified peptides, proteins, and post-translation modifications, but obviously not only the identifications, but also the quantitative results, which can be quantitative measures over many samples nowadays, and will belong to the main output of the MaxQuant software. And so this quantitative proteomics data will then typically be handed over to the PASEF software, where the statistical and bioinformatics analysis is done, where one can combine the data with protein annotation, where you have many visual tools to look into the data, and where you can also do multi-omics comparisons if you, for example, have done transcriptomics of the same samples. Then this would be the place where you can integrate these data types. So the MaxQuant workflow consists out of many algorithmic steps. So here you see what the workflow looks like for conventional LC-MS data, which does not have the ion mobility dimension included into the data. So basically that's the starting point for the developments that we did to accommodate the timsTOF data in MaxQuant. And so for each of these algorithmic steps, one can now think about, so is this going to be different for ion mobility enhanced data, or is it more or less the same and just needs some minor adaptations? And so there are actually only few steps which are going to be very different for ion mobility enhanced data, and most of them are quite similar, but obviously need adaptation because you talk different to different data type and data structure. As an example, for something that doesn't need much adaptation, you can think about the assemblies of peptides to proteins, so that step would not really know or not really care about how you actually acquired the raw data. While, for example, steps like detection of features, so what would be peaks in simple spectra, what would be three-dimensional objects in LC-MS data. So obviously this will be very different if you have now a new dimension added to your data. So that the feature detection, also the preparation of MSMS spectra, since they will also be coming from a higher dimensional space now, will also be quite different for this new type of data. And so here you just see the 3D features that I already mentioned that were kind of the unit or the main currency in LC-MS data without ion mobility. So here we were always talking already in every data analysis step about MS1 data as three-dimensional objects, because one deals with hills or mountains over the mass retention time plane. So intensity that is spanning over the mass retention time plane, which makes them three-dimensional objects. So here you see now a comparison between, on the left side, conventional LC-MS data, and on the right side of the table, the LC-TIMS ion mobility enhanced data. So as I already explained in the previous slide, we have a two-dimensional input space which is spanned by the mass, or mass divided by charge, and the retention time. And this is now going to be transformed into a three-dimensional input space. So to mass and retention time, there will be added the ion mobility index or the collision cross-section, or however you want to parameterize this new dimension. Then the features itself, they used to be three-dimensional objects because they used to live in a mass retention time intensity space, so that makes them three-dimensional objects. Now also these get the added dimension, the ion mobility, so they become four-dimensional objects. And so with that, there already obviously come problems with visualizing the data. So the three-dimensional data could be easily visualized, and probably many people know these kind of plots, how one visualizes LC-MS1 data, either as heat maps, where the x and y axis of the heat map are mass and retention time, and then you color code the intensity over this heat map. Or they can be easily shown as 3D surface plots, where you basically, that's essentially what we saw two slides back, where you display this mountain that is a peak in the LC-MS plane as a 3D object. So, but this is not possible anymore. So with this added dimension, we are running out of dimensions that we humans used to live in. So one has to think about ways to still be able to find out what's going on in the data by visual inspection. And so for this, we have also implemented in the MaxQuant software possibilities of projections, which means that you basically have to slice your data in one dimension. So you keep one dimension fixed, for example, the ion mobility index, and if you keep that fixed, then what you see then looks very much like like what conventional LCMS data looks like. But you could also fix any other dimension, like for example the retention time or the mass, and then do your conventional three-dimensional visualization on these other dimensions that are not kept fixed. And this you could, for example, center. So if you're interested in one particular feature, you could center this fixed dimension into the middle of this feature that you're interested in, and you can use the others to explore your space in the ways you are used to do it. So this is what you have to think about if you want to visualize the data. But how do we actually analyze the data? So we have what the problem is now. We have these three dimensions spanning this new space, including the ion mobility, and over that we have now some intensity, mass spec intensity profile in which we want to detect features, which in principle could be reduced to a problem of object recognition. For example, in fMRI data, one would have already developed object recognition algorithms in this kind of 3+1 dimensional data. However, since our other concern is not only to do good feature detection, but also to do this in a timely manner, so meaning in a way that is highly performant in terms of computation and leads to an answer in an acceptable time for the user, we try to use any regularities in the data to simplify the algorithms and actually use algorithms that are as simple as possible, but not simpler, as Weissman before me said. But that's actually a very good strategy to address this problem. And so what we do is, after a lot of smoothing and gridding of the data, so that it's smooth data on a regular cube spanned by these three dimensions, ion mobility, M over Z, and retention time. So after we've done that, to actually slice the data space, as you can see in this picture here, we slice it along the ion mobility index or cross-section axis. And the good thing is about this slicing direction of the data is what we get then is actually very similar to the LCMS data that we have been dealing with before. So if you now take some excerpt of one of these slices and look into it, it looks exactly like the data that we were dealing with before. And that's actually good news. So that's kind of the mathematician's approach, always reduce the unknown problem to a known problem that you have already solved, which we did by this. And we can now use in each of these slides the conventional MaxQuant algorithms to solve the problem of the feature detection. So then we have features. So as you can see in this picture here, then so what is color coded here are now isotope patterns or features found in one of these slices. And the next problem is then actually to put these. So here you again see some zoom into the detected features in one of these slices, and you see so the features that we detect here actually have irregular shapes, right? So it's not just rectangles that we take out of here, but it's really shapes that are following the features as they are in the data. And so once we have this for each slice, the next step is actually to put together these features that we found in separate slices. So here you see an example where ion mobility is increasing from the left to the right side, and you see now a feature appearing, growing, and disappearing. And so that's kind of a continuous process because of all the smoothing and regridding that we did prior to this feature detection step. It's actually a quite continuous process that we're going to find here. And then simple assembly or clustering algorithms will then be able to show to you what this three-dimensional, or this would be, you can imagine, kind of the surrounding or the surface that is actually embedding this four-dimensional feature that we recovered through this assembly process between the slices. So then we have the four-dimensional features. We also have to deal with the MSMS spectra, so they are just going to be basically projected onto a normal MSMS spectra as we know them, because for each of these MS1 features that we have, we know basically where to look for the PASEF data that it corresponds exactly to this MS1 feature. It just needs to be projected down again to this one-dimensional or two-dimensional data, and so the spectra that we get out of this will look like MSMS spectra as you know them from normal LC-MSMS data. And then we have all the parts together that we need for the further analysis of the data. So we have the MS1 features and we have the MSMS features. they can be essentially put into the conventional MaxQuant algorithms. I just want to, before I hand over to the next part, give you an overview of a few more interesting features or important features, because it's not just identification that is important, but most important in proteomics today is actually quantification. For this, we are implementing the matching between runs features that you might know from conventional MaxQuant algorithms, which basically identifies MS1 features and now actually these four-dimensional features which have not been sequenced by PASEF. So even though we have this big sequencing power with PASEF, this will actually further increase the space of MS1 features that you can use for quantification and will complete the quantification profiles over many samples. Here you just see an example, so you will actually see many more real data examples in the next part of the talk. Here, just to give you a taste, this is just the benchmark data of label-free quantification, where we basically know which of the proteins should be 1 to 1, 1 to 2, and 1 to 3. And so in this case, one can see that actually the label-free quantification. We also have isobaric labeling working already in the version that is released. And due to the time that seems to be proceeding faster than I was expecting, I will actually just mention another project that is still in the pipeline and soon to be released, and that the timsTOF PASEF will also benefit enormously from, which is the 2.0 version of the Andromeda search engine, which will have many interesting new features, like, for example, using the prediction of intensity of spectrum intensities in MSMS spectra from the peptide sequence, which can then be used to actually significantly increase the amount of MSMS spectra that can be identified from each LCMS run and also from each LCMS I-mobility run. So let me just skip over these interesting details, but just describe to you what you see here in these last 5 sentences. Trust me that it's working nicely. I just want to remind you again or summarize availability of what I've been talking about. So the timsTOF Pro support in MaxQuant is already done since version 1.623, which is freely downloadable from the MaxQuant download webpage. And if you want to find it, you just Google MaxQuant downloads, for example, and you will find the page. And this already includes the label-free quantification algorithm of MaxQuant and also the TMT quantification. So all of that you can do already. The match between runs that I was mentioning will be coming in the next release, which will be quite soon. I don't have the exact date, but that you don't need to wait a long time for this. The Linux support, so by the way, MaxQuant is now running fully under Linux. However, the file reading from Brooker file formats still needs a tiny tweaking, and that will also be in the next release that is coming out. So in the 1.623 that you can download now, it's actually not running on Linux. So it would have to be a Windows server. But there is also for this, you don't need to wait long. As opposed to Andromeda 2.0, although it will be very spectacular, it will actually take a little bit longer, and this you will be able to enjoy later this year. And with this, I'm finished with my part, and I hand over now to Andreas, who will talk about even more interesting things.
Hello, it's Andreas speaking. Since we now covered all the hardware stuff and all the software parts, how we analyze data, I want to talk about numbers and the performance we get with the timsTOF Pro in combination with the PASEF scan mode. And so here in the first experiment, we evaluated the performance of this combination with different ramp and accumulation times, and therefore we did the measurements with only 200 nanograms of HeLa digest in 120 minutes, and we performed quadruplicate experiments. And we found out that an accumulation ramp time of about 50 milliseconds yield the best performance with protein group identifications of nearly 6,000, and this converts to about 33,000 peptides on average. And here it is interesting to mention that the median peptide length of each peptide that was sequenced and identified is 15 amino acids, and the median protein sequence coverage sums up to about 13.4%. So in the next step, we also wanted to evaluate the quantitative performance of the label-free quantification algorithm of MaxQuant on the timsTOF Pro data, and we found out that the run-to-run reproducibility was super high with a Pearson correlation coefficient of 0.974, and also with a median coefficient of variation on protein group level of below 10%. And what's also really important to mention is that that the data completeness on the LFQ data in these quadruplicate experiments we performed was above 90%, and this is sort of the performance, or even better performance than you will get with data independent acquisition scan modi. In the next experiment, we wanted to evaluate the quantitative accuracy of the label-free quantification algorithm on the timsTOF Pro data, and here we performed a mixed proteome experiment where we mixed HeLa and E. coli in ratios of 1:1 and 1:4, and measured these samples again in 120 minutes and in quadruplicates. And after median subtraction, you can get these nice quantification plots, and you can so in total we identified here on 200 nanograms of input material more than 5,200 HeLa proteins and more than 1,200 E. coli proteins, and both species separate in the estimated ratio of 1:4, and they also separate in very nice, accurate protein clouds. So since this mass spectrometer, as Florian already told, is super fast and super sensitive, we thought it's time to perform a HeLa digest dilution experiment, and we started with 200 nanograms of HeLa and then diluted this amount twofold down to 3.125 nanograms. And astonishingly, we still were able to identify more than 1,800 protein groups from only 3.125 nanograms, which converts to about 10 HeLa cells. So this means that in theory, this machine seems to be set to go into really low sample amounts and into the analysis of really low cell amounts, cell populations, and maybe also into single-cell proteomics. So in the next step, we wanted to evaluate the sequencing speed and sensitivity of the machine in a more systematic manner, and therefore we used different sample input amounts varying from 100 nanograms to 50 and 10 nanograms, and also we varied the LC gradient time. So here you can see the output of the 100 nanogram of the 60-minute LC gradient measurements, and with only 10 nanograms, we identified more than 2,700 protein groups, and with the 100 nanogram, we identified more than 75% of the proteins we identified with double the sample amount and also double the LC gradient time. So this means we are sequencing so fast that we are still able to identify a huge portion of longer LC gradients. So then we went ahead and also decreased the LC gradient to 30 minutes, and astonishingly we were able to identify the same amount of protein groups, same number of protein groups in 30 minutes compared to 60 minutes, which again shows that we still don't reach the speed of identification with our actual sequencing speed. And then since since some time we have this this new LC in our laboratory, which is called the EvoSep1, and this the strength of this LC system is very low, are very small gradient times, and also the carryover is super low, and also the LC downtime is minimized in this case. The main issue is that when you perform measurements on other LC systems, you decrease the gradient time. In the end, your mass spectrometer is sitting longer around than it's actually measuring, because the overhead time is too large for these LCs, and this is not the case for the EvoSep, especially for the 5-minute, 10-minute, and 20-minute gradient. And here we try to push the sequencing speed to its limits by using the 5-minute LC gradient of the EvoSep, and here we identified more than 1,100 protein groups with an excellent reproducibility over 10 runs. Then we also evaluated the ability of the timsTOF Pro in combination with the PASEF scan mode in terms of deep proteome characterization, and therefore we used a basic reverse phase fractionation, which is automated on the spider fractionator by Proteomics, and here we fractionated 5 micrograms of Feeler digest into 24 fractions of approximately 200 nanograms, and measured them in 45-minute gradients on a 50 centimeter C18 column. And in total, we identified more than 9,300 protein groups in one day from minimal sample input amounts. And at this point, we asked ourselves, is there the numbers are super good, the sensitivity is super high, the sequencing speed is also super high, and now the question arose if we can also do biology with the setup. And since we are so fast and so sensitive, we thought it is a good idea to look into protein-protein interaction networks. And as we all know, if you want to create a protein-protein interaction network, for example, of yeast cells, you have to label the whole proteome and measure every protein to create nice interaction networks. And this would take or has taken in the past approximately 2 years, and we want to decrease this time. So in general, how do you create protein-protein interaction networks? Here you go ahead and create separate cell lines for each protein. You tag every protein that is encoded in the genome with the GFP, and you get this cell line, and you end up with 4,600 in terms of yeast. 4,600 cell lines, and then what you do is you lyse your cells, you fish for the GFP for your GFP tagged bait protein, and all specific interactors which can either be weak or can either be strong interactors are fished out by this immunoprecipitation. And then you perform a sample preparation, do the mass spectrometry on it, and then you do the data analysis to search for specific interactors, for stoichiometries of your complexes, and you can also calculate your cellular abundances of your proteins, but also your complexes, and then you end up with these huge interaction networks. So the issue itself is that you have to perform approximately 4,000 pull downs to create a representative network, and when you are super fast with state-of-the-art hardware, it would take 200 days of pure measurement time. And this is at this point you don't even take into account LC downtime and mass spectrometry issues. And here the aim is to evaluate the timsTOF Pro and PASEF for high sensitivity turbo interactomics. And therefore, we have a nice project which is led by a PhD student called André Michaelis in our laboratory, and he's optimizing with us together the whole workflow for yeast interactome studies. And in this case, we looked into 50 to 100 nanograms of pull-down digest, measured it into 23-minute LC gradients, and we investigated the whole protein complexes that are distributed across the full protein copy number range, ranging from 100 copy numbers up to 45,000 copy numbers. And on the right side, you can see the 4 representative complexes we especially looked at, and 3 of them are super low abundant. And in the end, we were able to find all complex members, all known complex members, with our setup in a quantitative manner. And on the left side, you can see enrichments of these network interaction members in a quantitative way, plotted against each other. So with this, we want to finish. Say thank you very much, especially to our whole department and the guys from Bruker who support us.
Well, thank you very much for the great presentation. Now we'll begin taking questions from our audience. Let's see what's in the queue. In a typical run of 10 versus 10 differential study of, let's say, cell lysate samples, how long does MQ take to give the final quantitation results using a typical chromatography ramp of 60 minutes? Here the time is counting from importing the raw files to obtaining the final results.
Yeah, I guess one could also rephrase the question: what is the extra factor that it takes between conventional LCMS runs and TIMS runs? And so I think we are at the moment at the factor of 3. So you have to maybe take 3 times more computation time to analyze timsTOF runs compared to a conventional, for example, Orbitrap style LCMS runs.
But this also depends on the computing power. So since MaxQuant is super parallelized, and when you have access to a lot of cores, it won't take much longer. So the point is, when you analyze one raw file, let's say. So one of the limiting steps is the de-isotoping and also the feature detection. And the time MaxQuant needs for these steps scales quite nicely linearly with the LC gradient time. So when you perform the analysis on one file for, let's say, 120 minutes, it takes approximately 16 hours. But when you have access to, let's say, in this case, it would be 20 files you have to analyze, and then let's say you have to access to 20 cores, then it would take approximately the same time as when you would analyze one file. So again, 16 to 18 hours for 20 files.
So which flow rates do you use when applying these very short LC gradients?
Yeah, so with the easy LC, we use 400 nanoliters as flow rates, and so with the EvoZap, these are standardized. It's 1.5 microliters.
And when a HeLa cell digest sample is running, is the 50 centimeter C18 column a commercial column? And what company is it?
So in this case, we use our in-house columns with the in-house pulled emitters packed with C18 beads from Dr. Meier. The inner diameter of the column is 75 microns.
And how are the precursors selected?
So I guess this question refers to the precursor algorithm that's implemented in the PASEF acquisition engine. And so in this case, the precursors are selected in the full dimensional space, meaning in immobility dimension, MRZ dimension, and the algorithm also takes into account the LC elution dimension, in a sense that precursors that are already sequenced are not going to be sequenced again. So there is a type of dynamic exclusion, and in the same way, precursors are ranked by their abundance, but the scheduling algorithm takes care that it maximizes the use of the immobility separation. So it always tries to put in as many precursors as possible based on the timsMS survey scan.
Is it possible to use timsTOF Pro for metabolomics experiments?
I think from our experience, it should be possible, but this study we did not perform experiments in this direction.
Where can I get the latest MaxQuant version?
So I didn't provide the link, but the best way to get the latest MaxQuant version is really to go to Google and type MaxQuant download. It will the first hit will bring you to the download page, and it's actually only the latest MaxQuant version which you can always download.
How are the CCS values calibrated?
So we, for the study shown here, we use the Agilent tune mix, the phosphocene derivatives, and they are unknown CCS values in the literature, and we can readily calibrate the TIMS dimension to the known 1/K0 values from the literature. And if you want further details, I can actually recommend a review from Mel Park and colleagues where they describe this procedure in full detail.
Can you use lock masses / internal calibration with the timsTOF Pro?
So in principle, one could think of things like that, but the beauty is that if you actually use MaxQuant for the data analysis, so it will not require any of that. So it will calibrate on internal calibrants from your sample.
So what is your experience with the robustness of the instrument? How often have you had to clean the quadrupole or collision cell?
Yeah, so we never opened the machine to clean any parts except for the glass capillary itself. And therefore, you have to shut down the vacuum pumps, which takes approximately 1 hour. You clean the glass capillary with a routine workflow, which again takes about 1 hour. You put it back in and turn on the vacuum pumps again, and you get a nice vacuum after about 4 to 6 hours. So in total, you have a downtime from 6 to 8 hours until you can start to measure again.
Is the manuscript already published?
So you can find the manuscript on bioRxiv. We already uploaded it, and the easiest way is to Google online PASEF or online Parallel Accumulation, and then Mann, Meier, Brunner. And then you will find it. That's the first hit.
Okay. I think we have time for a couple more questions. So have you tested the peptide mapping of pure protein, say 150 kilodalton antibody? Were you able to achieve 100% sequence coverage?
Yeah, very interesting question. The short answer is no. we would guess that the very fast sequencing speed of the PASEF will actually allow to get close to these numbers, but as I said, we haven't tested this far.
Okay, and our last question is, what is the impact of high charged peptides in timsTOF Pro?
So they can be separated by their ion mobility, just as their lower charged counterparts. Typically, we are working with cryptic digests, so most of our peptides will have charge 2 or charge 3, and we don't have much experience with longer peptides.
All right, well, that's all the time we have. Thank you again, Andreas, Florian, and Jürgen, for your fascinating presentation. And thank you, participants, for being such a great audience. Be sure to check CNEN or CNN Online for information on the next edition of CNN webinars. Thank you, On24, for technology and production services. And thank you, Bruker, for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Elaine Lim. Goodbye.